My name is Benjamin Joseph, and this is my presentation on controlling lanternfly damage using magnetic fields. First of all, what are lanternflies? Lanternflies, otherwise known as Lycorma delicatula, are a dangerous invasive species native to China that were first spotted in Pennsylvania in September of 2014. The reason why lanternflies are so dangerous is because of their ability to cause damage to plants through sap oozing, wilting, dieback, and excreting a substance that leads to the growth of black mold. Additionally, lanternflies are known as hitchhikers and will attach to anything they can get a hold of, and use it as a method to get around and plant their eggs. These insects are so problematic that the instructions for when they are found or when their eggs are found is to eliminate them as soon as possible. The focus of my experiment is to try to reduce the amount of damage that lanternflies cause by using magnetoreception. Magnetoreception is a sense that enables some animals to detect magnetic fields. They use this ability primarily to detect Earth's magnetic field so they can determine their location, direction, and their altitude. This ability typically plays a major role in the navigation of various animals. Various researchers have performed research on certain insects in order to test if they are specifically capable of that sense. Examples of this is the study Can Altered Magnetic Fields Affect the Foraging Behavior of Ants, performed by Pereira et al., where they had found that ants, when in the presence of a magnetic field, instead of walking straight to a resource and then back, they instead began to turn and take longer paths coming back and forth. A similar experiment to this was the study A Magnetic Compass Aids Monarch Butterfly Migration, performed by Guerra et al., where they had found that monarch butterflies were capable of magnetoreception due to their antennae, which have light-based magnetoreceptors that are vital to their internal compass. The focus of my study is to determine if this sense is present in lanternflies, and then see if this sense can be manipulated in order to restrain the movement of lanternflies and keep them away from the different plant life that they damage. This brings me to my research question. To what extent can magnetic fields manipulate the movement of spotted lanternflies? My hypothesis for the study was that if lanternflies are capable of magnetoreception, then their movement should be able to be manipulated because they normally incorporate the sense into their navigation. In order to properly answer my research question, my experiment utilized a Helmholtz coil, a Y-maze, painted lady butterfly larva, also known as Vanessa cardui, and Vanessa cardui culture medium. Due to lanternflies being known for their ability to attach to various objects and then plant their eggs, they could not be used for this experiment. As a replacement for spotted lanternflies, painted lady butterflies were used instead. The reason for this is because painted lady butterflies and spotted lanternflies share the same class, so they would be a close enough alternative. So any results that were determined about painted lady butterflies were assumed to be the same for spotted lanternflies. The Helmholtz coil was made by taking two styrofoam rings that had an outer diameter of 37 centimeters and then separating the two of them by 18.5 centimeters. I then took 14 AWG wire and coiled it around the two rings 20 times in order to create a strong magnetic field that was 60 microtesla. The coil was then used to test if the presence of an artificial magnetic field would cause any significant changes in movement. The Y-maze was created using two wooden pieces that were elevated and positioned in a way so that the food on each side were an equal distance away from the starting position of the larva. The setup was based on the study performed by Pereira et al. The setup consisted of placing culture medium on both sides of the Y-maze and then placing the larva in a smaller area that is right before the splitting of the pathways. The procedure for this experiment was to place culture medium on both sides of the Y-maze with the Helmholtz coil on only one side. The Helmholtz coil was then turned on for half of the trials, while for the other half it would be turned off. Then we had to release the larva into the Y-maze and observe which direction that it chose, and how long it took to get there. This was repeated 12 times, the Helmholtz coil being turned on for 6, and then turned off for the other 6. This was to ensure that any results that were determined was as a result of the experiment and not because of outside influences. Each larva was given a maximum time of 2 minutes and 30 seconds to reach the food. The completion of the experiment and the results that were gathered from the study reveals the tendency of painted lady butterfly larvae. The process of determining any significant patterns were done so by tracking the time that the caterpillars took, as well as observing which direction that was chosen each time. The purpose for recording the time in this study was to determine if the caterpillar had been moving at a different rate when the magnetic field was a part of their surroundings. What was seen was that the time that was required for both groups were fairly similar to one another. With the exception of the outliers seen in the results that were included in the magnetic field, most trials had a maximum time of 2 minutes and 30 seconds, or very close to that time. This was used as a method in order to determine if the addition of a magnetic field would affect the movement of a painted lady butterfly larva in a particular way. By observing which direction that the caterpillar took, I was able to determine if there were any specific patterns that correlated to an effect caused by the magnetic field. What was observed through the experiment was that when the magnetic field was on, the caterpillar went forwards, which is where the magnetic field was placed. However, when the magnetic field was off, the caterpillar tended to move towards the left. What can be inferred from this result is that the larva in the presence of a magnetic field will head to where the magnetic field is stronger. The finding of this experiment was significant because it determines whether or not spotted lanternflies might be affected by magnetic fields. From this result, it could be inferred that there is a correlation between the movement of the larva and how they move closer to the source of a magnetic field. However, there were some limitations to the study. For example, the magnetic field that was used in this study could have been a static magnetic field, which would allow for the entire field to be the same strength. Another limitation of this experiment was that the caterpillars and how they refused to move in certain situations. One final limitation to the study was that during the time of the experiment, the larva was in a random stage of their development, and so that could have affected the results that were found in the experiment. What could be improved in the future studies is instead using a static magnetic field to make sure that the fields have a constant strength, significantly reducing the distance that the caterpillar starts from the resource in order to alleviate the problem of their laziness, and to keep track of the stages of their development so that it doesn't isn't a significant problem with the results. These changes are vital to the study because they will eventually lead to an answer for lanternflies and whether or not they can whether they have magnetoreception and whether magnetoreception is a sufficient answer to the problem that lanternflies are in the environment. And these are the resources that I used to base my study. Thank you.